Hi, everyone. So what I'm going to talk about today is actually research that has not been conducted, but research that I think could be of value to this community. So an outline of my presentation, I just want to describe what I call a roadmap. I want to introduce you to the topic of spatial interaction models, which is an area of specialization of my research group here at the University of Georgia. Some models that I think we could develop for JEV spread in Australia. I think it would be useful for us to retrospectively conduct an analysis of West Nile virus in North America. And then I think lessons learned from those two activities could actually be quite useful in developing a model that we might use in advance of any introduction of JEV to North America to prioritize, for instance, surveillance, interception, and control activities. So we've already seen something like this already, the life cycle of Japanese encephalitis virus. It was interesting to me that when our speakers from Sunport presented on what's known and unknown about JEV in Australia, so many of the unknowns are in fact ecological factors. As an ecologist, I'm not surprised. There's much we don't understand about the ecology of wild animals. And of course, we're changing that ecology all the time as we modify the world that we live in. So we know that the JEV life cycle includes various QLX species, but possibly other competent vectors, wildlife reservoirs, which may or may not be the same between Southeast Asia and North America, a spatial interaction between those locations that are amplifying, whether that's because they are built environments, for instance, pork producers, or wild environments where birds can be amplifying or feral pigs can be amplifying. And of course, many of our sentinel species are dead-end hosts. But this looks an awful lot like a virus that we're quite familiar with in North America, where we have amplifiers both in controlled populations and in wild populations, dead-end hosts that function as sentinels, mosquito species that are part of that amplification, and of course, all of these subject to seasonal variations in weather and other environmental triggers. So here's what I think we need to do. The big unknown is what would JEV spread in North America look like? I think potentially we could develop a spatial interaction model, which could then be used for scenario analysis. So for instance, the last speaker suggested that the probability of introduction could be roughly the same between Mediterranean California and eastern deciduous forests. But the subsequent spread of JEV in North America might be quite different based on, for instance, the distribution of vertebrate bird populations, breeding bird colonies, and of course, feral swine. I think we need a number of ingredients. One is a JEV importation model. The last speaker gave us a very detailed explanation of what that looks like and has informed us that improvements to that model are ongoing. So I'm eager to see the outcomes of that result. I would like to see a quantitative analysis, spatially distributed in North America, of where we could expect those introductions to begin. I think we could learn a lot by better understanding the spread of JEV in Australia. I'd like to understand what the roles of topography, climate, and the ecological community are in preventing spread. Now, the picture that is developing in my mind for what happened in Australia is that introduction happened a couple of years ago and spread throughout the wildlife population, largely throughout the country, and probably what in my line of work we would call near critical transmission. So it probably spread undetected in wild animal populations. And so, of course, once there were the triggers for the mosquito population to exceed a threshold, causing there to be supercritical transmission and spillover into domesticated pig populations, it was only then that we finally saw it. So we had kind of a two-stage process with a sleeper epidemic, which was then followed by an environmental trigger, which caused it to outbreak simultaneously everywhere. Whether or not we can use a spatial interaction model to disentangle those different pieces, I think, is a very interesting question for further scientific research. I do, of course, understand that most of the data we have is a significantly lagging indicator of transmission that happened either on pig farms or in the wild environment. I think that would inform a JEV spread model in North America, but I think the conditions in North America are also quite different. Different players, possibly different spatial distribution of amplifying spread versus migration. The picture that's forming in my mind is that we need to think of the species that are spreading the virus and the species that are amplifying the virus as different. So when those are co-located is really important. And of course, that's seasonably variable. As is, of course, the mosquito population dynamics. The role of wild pigs in North America, I suspect, would be somewhat different, although perhaps informed by our understanding of the role of wild pigs in Australia. And of course, mosquito species so the JEV is a promiscuous species that can, virus species that can associate, we believe, with multiple vector species. Mosquitoes worldwide are distributed with relatively limited ranges, of course, with exceptions that we're quite familiar with. And so some kind of statistical approach to anticipating which mosquito species might be competent in North America, I think would be a good first step, followed by, I would hope, challenge experiments to verify those predictions. All right, so what would we do with all of this information? One of the things that we work on is spatial interaction models. Most models of infectious diseases are not of this kind. They're considered to be compartmental models. Mathematically, those are systems of differential equations which describe how different host populations amplify the pathogen of interest and how transmission occurs, what those routes of infection are. And mathematically, often what we do is we derive criteria then to determine whether or not a particular parasite or pathogen could invade a population of interest. Unfortunately, those compartmental models are really only useful in local contexts, what theoretical biologists call well-mixed. And by well-mixed, what we mean is that the spatial distance between two individuals actually is relatively negligible to ongoing transmission. Compartmental models like this might be useful in the context of thinking about transmission within a specific pig herd or in a localized environment. I think in the United States, that would be something like the spatial extent of a county or a multi-county area. But by the time we're talking about transmission in states or spread across the country, compartmental models like this, which are well understood, are pretty useless. Spatial interaction models, by contrast, think about locations in space as having their own properties. They're amplifying or not amplifying based on properties of that location. The host species and vector species that are present, the times of the year that they're present, this kind of thing. And then we think that there's a redistribution kernel, which has to do with the propensity for movement from one location to another. And all of the spatial interaction modeling is about trying to figure out what that kernel is like. Now, this kernel is not something that's directly measurable, but what we do is we statistically infer what that kernel looks like from the spatial spread of a pathogen across a landscape, together with information about the properties of the connection between two locations. So for instance, whether there's vehicular or airline traffic between those locations, whether there's a migratory pathway for wild birds between those locations, as well as the properties at those locations, like, for instance, the concentration of pig farms or the presence or absence of wild pigs. And then we estimate what this statistical quantity is. And so usually we think from one time step to the next that this is represented as a probability. So it's a number that ranges from 0 to 1. And using biostatistical tools, well, we can estimate what relative risk factors are of various covariates of interest. So for instance, were we to do this with information on the spread of JEV in Australia, I think we might be able to measure the effect of elevation as a natural barrier to the movement of JEV. Well, that could be quite relevant and perhaps transferable to the North American context, where, for instance, we have the Rocky Mountains and the Sierras that are separating the California Mediterranean climate from, say, for instance, the Central Plains. We have used this modeling framework in real time in the past for a number of different projects. One of these was in support of the NIH Midas network during the response to the West Africa Ebola epidemic. I know that Ebola and JEV are very different pathogens, but at the gross macroscopic scale, I hope I don't have to do this. I don't. I hope that doesn't speak to the statistical and technical incompetence of our group. the question at hand was, what are the likely paths of the spread of Ebola throughout West Africa? This was at a time that the US government was supporting standing up 17 Ebola treatment units throughout the country of Liberia. We wanted to stand up those units in advance of the traveling wave of Ebola. And so anticipating where those would be in the heterogeneous space of a population which differed tremendously in terms of things like population density, compliance with recommendations, infection control, health infrastructure, and so forth. All right, so how does that look like, Japanese encephalitis virus? Well, you still have locations that are either infected or uninfected that have the acquisition of the pathogen at a particular point in time, and gross covariates such as population density or the setup of the interception and barriers to prevent the continued spread. We actually pioneered this approach in the context of white nose syndrome, an emerging fungal pathogen of bats back about 10 years ago. And more recently, we've deployed it in cooperation with the Division of Global Migration and Quarantine at the CDC, which, for instance, supported 2 years ago the DRC Ministry of Health in their ongoing response to Ebola virus in Central Africa. We also use this in the United States at the county scale to understand the spread of SARS-CoV-2 in the summer of 2020. This is the same kind of thing that I could imagine we would do for JEV. We developed first an importation model. This was done in coordination with the Division of Global Migration and Quarantine, much like the model that Natalia just spoke about. This then is the initial condition for a subsequent county-to-county spread model, which allows for both diffusive local spread as well as long-distance spread through what we sometimes think of as teleconnections, those connections between two locations that are based on, for instance, vehicle traffic and so forth. So I think we could do this with information from Australia if our collaborators are able to help us in obtaining the right kinds of data. I don't think this would tell us how it spread throughout Australia initially. So my idea for what happened in Australia was that there was this kind of sleeper spread that happened throughout a number of different regions simultaneously. It wasn't observed. It spilled over into pig populations and maybe also wild populations, for instance, breeding bird colonies. And then these were like the epicenters of local spread that was diffusive. It's that second piece, the sort of local spread, for instance, up and down the Murray-Darling basin, that I think we might be able to infer with the right kind of information. This would then allow us to assess the relative roles of things like topography, relative wind direction, concentrations of animal species, and so forth in contributing to spread. This would require that we obtain information on the inferred time of initial transmission at different locations, covariates at whatever granularity those could be made available. And then we could use our spatial interaction model and the statistical method of maximum likelihood to try and infer what that dispersal kernel is that I was talking about before. So also, I think we can do the same thing with West Nile virus from our surveillance in 99 through 02, during which time West Nile virus spread across the US. Well, actually, I don't think it spread across the US. What I think it did was a kind of sawtooth pattern where it first spread down the eastern seaboard. The mixing of migratory birds during overwintering brought it back first to the Midwestern United States, then to the Rocky Mountain area, and then eventually it spread over into the Pacific coast. I think we can do the same kind of thing and infer what some of those gross features of the North American geography might be to influence the spread of JEV in the United States. This would, of course, be informed by a number of different covariates, which would include things like the distributions of the dominant mosquito vectors, the distributions of amplifying bird species. In the case of West Nile, this might be, for instance, cardinals or crows, which differ, of course, as to whether they are amplifiers, but not particularly symptomatic, or symptomatic sentinel species that we could monitor. Time-varying covariates like temperature, precipitation, wind speed, and direction. These are the kinds of things that we would use as covariates. And the outcome would be something like a spatial interaction model parameterized for West Nile virus, which we might then modify in the context of what we could learn about JEV in Australia to do some scenario analysis that would then be useful for anticipating potential spread of JEV should it be introduced. This is going to require that we obtain some additional information. So for instance, I think we've already seen information about this and other reviews of the ecology of JEV. Obviously, these reviews are not exhaustive because they're limited to the global experience with JEV. And we're talking about translating that into a novel ecological context. My group has some data analytic experience with trying to do that. In the course of the Zika virus pandemic, one of the things that we did was a data-driven horizon scan looking at potential competent vectors for Zika virus in North America. This was prior to the opportunity to do challenge experiments or to observe the natural spread of Zika in the continental US. Information on bird species is readily available from a number of digital platforms and collaborators, such as the Cornell Laboratory of Ornithology. So I think those data could be obtained. Location of swine operations in the US could be quite useful. I think this would probably not be a covariate looking at amplification so much, but maybe information that we could layer over our final model results to help us understand where we would expect the American swine herd to be first affected. And that might help us with things like prioritizing surveillance and interception, or as our Australian colleagues suggested, locating mosquito control in the vicinity of swine operations. Here's where I think much of the amplification, much of the resident amplification in North America is likely to occur. One of the ecologically interesting things to me is that I think we need to make a distinction between those ecological communities that are actually amplifying the virus and maintaining it on the landscape and those that are moving it around. And the relative importance of these two things, I think, remains to be quantified. And this is one of those ways in which I think a retrospective model of West Nile virus could be useful to us. So some questions that then could be addressed are these: how is JEV likely to spread in North America from given importation points? If we use a probabilistic model of importation, then we can think about probabilistically what are the relevant scenarios for spread in North America. What can multi-scale modeling add to this picture? Should we do some compartmental modeling? Natalia was arguing about the relative probability of an introduction resulting in persistence and endemicity in local populations, for instance, in the vicinity of airports and seaports. I think that we can actually bring some of our technical expertise to answering that question. There is theory on how you answer those kinds of questions. What prevention, interception, and response programs would be most effective? Can we, in fact, optimize our policies for effectiveness? And then what are the geographical areas of greatest potential impact to the swine industry? What mitigating effects can optimized policies have on these impacts? And I think this is important because depending on what kinds of information we need, like we might do different things in different places. Some places might be really important for rapid detection. Other places are going to be really important for ongoing surveillance. And yet other places and activities are going to be important for damage mitigation. This slide is just to illustrate one of the things that you can do with a spatial interaction model. This is an algorithm that I developed together with colleagues at NC State. These colleagues study what's called personalized medicine. So personalized medicine is an innovative part of clinical practice where what you do is you collect data on an individual as disease progression occurs. Now, anybody who's taken biostatistics knows you can't do statistics on an N of 1. Well, actually, that's not true. If you collect repeated pieces of information and you have a structured model about the different pathways of progression, you can actually learn things statistically and then optimize your intervention, your treatment regime. Well, one of the things we realized is that the progression of an epidemic in space is a little bit like the progression of a disease in a person. And so all of this work that has gone into data analytics for personalized medicine could actually be harnessed for thinking about personalized interventions in an epidemic on a landscape. And we showed mathematically how you actually go about doing that. And we derived this algorithm for that purpose. Now, this algorithm is about optimal interventions. Are we actually going to be able to stem the tide of something like JEV? I don't know. But there's also a problem for optimal surveillance so that you can extract the maximal amount of information as a disease is progressing, and then at least adopt protective local measures. Well, what do we need to do this? There's a number of different data sources. One of the things I'm excited about this symposium is that we're getting a lot of the stakeholders and data holders together in the same place. I hope tomorrow we're going to be able to brainstorm about existing data, how it is that those data might be harnessed in a safe and secure way to maximize our collective benefit from that information. But then we'll also identify pieces of information, data that we don't have right now, and maybe could be prioritized for collection. And then there's a number of things that I think we should do. What my group brings to the table is the know-how and architecture for fitting these models, but we can't do it alone. There's too much expertise that's distributed around the world and information sources, as well as needs for information, right? So what the use case of such models is. So it's my hope that we might find a group of people that are interested in working on such things together. What might we do with such a multi-model architecture, and why would it be useful? I think that the value proposition speaks for itself. I think it is to prioritize locations for surveillance, interception, and control. All of these things are costly, but unmitigated damages to the industry could be hugely costly as well. And so the optimal allocation of resources, both in advance of and in response to any future introduction, is something that I think really could be informed by a model developed like this. Thank you.
Questions for John?
Hi, Megan Niederwerder from the Swine Health Information Center, everyone online. So I'm curious about your take on the ability to take what we've learned from West Nile virus and apply it to JEV. And I think one of the aspects that you go is, well, the species that we first identified West Nile in was wildlife, birds. Likely the species we first identified JEV in is commercial pigs or piggeries as Australia did. But Australia has also gone through both West Nile and JEV. So is there certain things that we've been able to identify based on Australians' experience that has differentiated their experience with West Nile and JEV that we could apply to the US, looking at our West Nile experience? Or were they similar? Were they different? What's your take on that?
Wow, that's a great and multifaceted question.
Sorry.
I think I don't have an opinion about where we're first likely to detect JEV. I think that has to do with things like where it's first introduced, what impacts it has either in wildlife or domesticated animals, as well as things that may change between now and the time of introduction, perhaps as a result of coordinated interest by groups such as this. As to the question of, well, and that's actually one of the reasons why I think we need to do an analysis like this, because it might tell us where and how to look, right? In terms of the lessons learned about West Nile, the difference between West Nile virus and JEV in Australia, I think that's where we would need to turn to some of our colleagues from the Australian context to draw up for us what the similarities and differences were. I don't know those myself.
Can I maybe just add a comment on that? And it's actually been a different scenario with West Nile virus, Kunjun in Australia. We've had sporadic cases, like one every, couple of years or something like that. So it hasn't been the establishment. And transmission like there was in the US. So it would be interesting to ask the Australians if there is any lessons, but it's a very different situation there.
Okay, oh, that's good to know though, that, you know, they're probably because of the different strain that they were exposed to.
Yeah, Kunjun, and there's a lot of Murray Valley is much more widely transmitted. So whether that's impacting is, yeah, it's interesting, but only a few sporadic cases of
Okay.
Kunjun.
Thank you.
I think the maps that we're using, this is David Reeves, a consulting swine veterinarian, but I think the maps that we're using on feral swine, the ones that I've seen presented today, are very outdated. I saw a presentation recently where in the Canadian plains, they had an absolute explosion of feral swine all across the plains, and it's become a huge problem. So I think in consideration for what you're doing and what you're talking about doing too, John, I think there's a need to update those maps for all of North America and not just look at what's happening across the southern part of the U.S.
Hey, Steve Reckham, USDA, SBS Health. Kind of bridging on that, just Dr. Jake, for your experience, I'm wondering what the value of getting above a decision threshold, right? We can get great data, we can kind of do the specific studies and figure this out. But if we've got kind of fuzzy enough numbers, even if the maps for the feral swine aren't great, but it's enough to kind of get us over that decision threshold, I'm just kind of wondering, and maybe this is more kind of asking you to speak about that, the value of specific information versus extrapolating, you know, from a layer or two away, and kind of where the goal needs to be set to how certain we have to be and how certain about the likelihood and the impact we're kind of looking for.
Right, and so I think this is actually one reason why a meeting like this is so important. There's a difference between the academic interest in a question like this, which is what we often find in universities, and the need for decision support and response. So what is the action space, and what are the pieces of information that would trigger a response of different kinds? And perhaps the granularity of the information you need is not the same as what you would need for a thorough understanding of the ecology or epidemiology of such a phenomenon. This is actually the reason for holding a meeting like this, and also the reason for why here at UGA we started the Global Infectious Disease Intelligence Consortium. I think that we often have conducted our research not in the context of particular use cases, and that it's done not in the context of collaboration with those industry partners, and outlining what those needs are, what those use cases are, what information would trigger actions by different stakeholders is exactly a part of this meeting this week. Tomorrow, we will have two sort of collective brainstorming sessions. And I'm hoping one of the things we can do is the collective brain dump and get it all onto a single whiteboard or a single piece of paper. What are the information needs or desires of different organizations or stakeholders? Please, Paul.
Paul Sundberg, Swine Health Information Center. And I think that's exactly the point of what's academically interesting and what is actionable within the industry or government or response. And I'll offer a description. When I heard your description of spatial interactions and spatial modeling, and you're bringing up the interest of elevation, the effect of elevation on the ability to spread JEV via mosquitoes or vectors. That very well may be academically interesting, but does this spatial modeling take into account the probability of human spread of mosquitoes, these vectors, once JEV gets into the US? It seems to me, the likelihood that we would be transmitting competent mosquitoes across the mountains in our cars every day would be a much greater factor in the spread of JEV than the elevation of the mountains keeping the mosquitoes from being able to fly from Nebraska to Nevada.
That's a great question. And I want to answer it in a quasi-technical way. So there's two philosophies of biological modeling. One is you go out and find all of the things that you know to be relevant and important. You measure them individually, And then you add them all up in a complicated computer simulation or mathematical model and say, if we put all of these ingredients together, here's what's likely to happen. Another approach, so that's sort of like the bottom-up approach, right? You collect all the pieces and put them together. Another approach says, let's start as coarse-grained as we can and envision the things that might be important, and then try and estimate statistically what their impact is. I personally believe that that bottom-up approach is very poorly validated. So very rarely are we able actually to discern what the synergistic effects of multiple factors are when we add them in that way. And partly that's due to model misspecification, partly it's due to measurement area. We saw this often in COVID-19, for instance, but people would measure all of these different things, the amount of spread on a city block, how kids mix in schools, and throw it all into a big model. What our spatial interaction models do that's different is they say there's probably a bunch of things that are important, but we don't know how to measure the relative importance of, say, the movement of birds along flyways or the movement of mosquitoes in people's cars, right? But what we do is we can collect covariates that we think are pertinent to those things, and then statistically partition the time at which an infection arrived at a particular location based on all the possible paths and the covariates that would have been encountered along those paths. So one of the things that we do is we look at vehicular traffic pairwise among all counties. And so if in fact the movement of mosquitoes over a mountain range in people's cars is really much more important than the movement up and down flyways, then our statistical model will significantly load onto that covariate rather than onto a covariate that has to do with the movement of migratory birds. The downside of such a model is it's not strictly mechanistic. We sometimes think of this as quasi-mechanistic in that we have mechanisms in the model, right? So those mechanisms include, we have mechanistic covariates that we have a plausible mechanistic explanation for, but they're not modeled in a way that's based on the specific understanding of how that works. So then we also don't have a variable that we can turn off. Let's suppose that we implemented a biosecurity measure that required, I don't know, some kind of treatment of every car that goes over the Rocky Mountains. What would be the effect? There's no way of asking that counterfactual with a model like this. But what it can do is help us to anticipate what the time of arrival would be, the likely paths that the epidemic would take throughout a geographic space. And to the extent that we have direct interventions as part of those data to which we're fitting our model, we can measure the effectiveness of those. So for instance, border interceptions was something that we have included in most of our Ebola models. One of the great things about a spatial interaction model like this is if we build it in advance and then JEV should arrive, we've got a baseline model, and as we collect data, we can update our existing model with new information as it comes in.
Thank you, John. Are there any other questions? All right. Okay, thank you very much, John.